So, today we will discuss T cell activation. It is a very important topic because T cell activation plays very important roles in the generation and sustenance of an immune response. It is also, it has clinical manifestations because, for example, during transplants, transplant surgeries and all, you want to suppress the T cell activation, and therefore immunosuppressants are used. On the other hand, with vaccines and all, you use adjuvants so that you can enhance T cell activation. So, these are very important aspects, and that is something that we need to understand. Okay. So, we will start off with the basics first. What is shown over here is a T cell with the T cell receptor. Remember, specificity for the T cell is via the T cell receptor, which recognizes the cognate MHC peptide over here. So, here you have an antigen presenting cell presenting a particular peptide on its MHC. This particular MHC peptide complex is recognized by T cells. By the T cell receptor, and it leads to T cell activation. So, what exactly happens? So, once T cells are activated, they produce IL-2 and other cytokines. IL-2 is an important one because IL-2 is the autocrine T cell growth factor. So, it is produced by T cells and it helps them proliferate. And that is and this process is also important for the killing of target cells by cytotoxic T lymphocytes, and we will discuss this in a little bit greater detail in the subsequent slides. Okay, so this is just to tell you a little bit about Cd4 and Cd8 function. Here what we have shown is a dendritic cell, which is an antigen presenting cell. It is presenting MHC class 2, and here is a Cd4 T cell with a cognate T cell receptor. So, upon activation, what happens is T cells, Cd4 positive T cells are helper cells. So, they help macrophages, they help B cells, and this leads to proliferation and differentiation of the Cd4 helper cells into Th1, Th2 types. These are the primary types, results in production of cytokines, so on. On the other hand, the Cd8 positive T cells differentiate from Cd8 positive T cells into CTL. So, this is a very important point. So, what happens upon activation is that these Cd8 positive T cells differentiate into CTLs. So, the CD8s differentiate into CTLs. So, there is a difference between CD8 which cannot kill on their own, but CD8s after differentiation become CTLs which can kill. So, it is a very important aspect, and these kill infected cells and tumor infected cells and T cells and tumor cells. So, this is a little bit more about CD4 positive T cells. These are helper T cells, as I mentioned. They activate macrophages, they modulate B cell signaling, and they differentiate into two major subsets. The Th1s, which are more pro-inflammatory, and that is because they produce lots of IL-2 interferon gamma, or Th2, which produce IL-4, IL-5, IL-13, which sort of reduce inflammation. So, the mechanisms by which Th1 and Th2 differentiate are well studied. So, what happens in case of Th1, this occurs in the presence of IL-12, which is produced by primarily by the antigen presenting cells. Macrophages are very good producers of IL-12, and this increases IL-12 increases interferon gamma via STAT4, and it leads to increase in STAT1 and production of Tbet transcription factor. So, one of the important signatures of Th1 positive cells is Tbet, and Tbet is very important because in the absence of Tbet you do not get the Th1 types. Now, similarly in Th2 development, it occurs in the presence of IL-4, it results in activation of STAT6, and it increases the transcription factor GATA3. So, you can see there are two important transcription factors that play roles here, Tbet for Th1 and GATA3 for Th2. Okay. Now, for Cd8 positive T cells, so I said this, these differentiate into CTLs now, which kill. Now, how is it, what is the difference between the naive CD8s and the CTLs? What gives CTLs the property to kill? And the reason for this is the CTLs have molecules known as perforins, and perforins make holes in the membranes of the target cells. and then through these holes you have molecules that get in, one of which is granzymes, and these are serine proteases which are capable of activating caspase. So they initiate the death cascade, and then you have fast facile signaling which results in apoptosis. So you have different mechanisms involved over here, and CTLs can kill because of these important molecules: perforins which make the holes, granzymes which are the proteases which activate the death pathways, and then fast facile pathways. We will be discussing fast facile and the later death pathways in subsequent slides, but for now I think this is a fairly reasonable introduction into the importance of T cell activation of CD8s which turn them into CTLs. Now it is also important to remember that there's a effector phase in which CD8 positive T cells divide several times. So once you have initiation of the immune response, these cells divide incredibly fast, and they increase by in some cases 50,000 fold in number. Now if you have that many T cells, activated T cells, it is bound to cause problems in the body, and that is why we have a system by which there is a contraction phase. So you have an effector phase which results in increase in the numbers of the CD8 and CTLs, and then you have a contraction phase in which most of these cells die and they leave behind a small number of memory T cells. So this is a very important aspect about T cell activation. You need to activate T cells, but after activation you would need to bring the numbers down or lower the activation, because if you do not, then if the host has to constantly be in the presence of huge amounts of cytokines and these activated T cells, it causes problems what is known as immunopathology. So this is again this is an important aspect, and so in T cell activation we need to learn both about activation as well as bringing down of responses. Okay. So now the other important point that needs to be emphasized is that the CD8 positive T cells are required to generate an effective CD8 response. The CD4s are required to generate an effective CD8 response. So if you see AIDS patient, the CD4 numbers are lowered, but the CD8s are there, but however the CD8 function in these AIDS patients is compromised. It is compromised because in the absence of appropriate CD4 help, they do not show proper function. And that is again tells you about the importance of the CD4s as master regulators of the immune response, and that is why they help macrophages, they help B cells, they help CD8s and overall orchestrate this immune response, and and this is manifested at and can be seen in case of AIDS patient who have reduced number of CD4, as a result of which their immune system is compromised and they are more susceptible to infections by opportunistic organisms that reside in our body. Okay. This is an important aspect. So why is it important to study T cell activation? So if you see abnormally enhanced T cell activation is observed in autoimmune diseases such as multiple sclerosis, rheumatoid arthritis, diabetes mellitus, etc. On the other hand, reduced T cell activation leads to increased susceptibility of the host to infections, microbes and tumors. A good example of that is AIDS and the HIV infection. Now importantly, suppression of T cell activation is required for organ transplants to be successful. So this is where while we were discussing MHC, and you will recall that the way Medaver got into scientific research was to help World War II pilots be able to survive skin burns, and that is where how he got interested into scientific research. But for skin grafts, in order for skin grafts to be successful, you would need to do grafts, and how do you achieve grafts that have a higher rate of success? So not only do you need to match the MHC, but you also need to suppress the endogenous T cell so that you don't reject the graft very quickly. and you have to give a chance for the graft to be successful, and therefore immunosuppressants are used. So cyclosporine, for example, something that we will study, is used, and cyclosporine and cyclosporine like molecules are used. Now, as I mentioned, for vaccines to be on the other hand, for vaccines to be successful, you need to boost immune responses, and you need to find out what are the mechanisms by which T cell function can be increased. So these are examples to illustrate the importance of studying T cell activation. Okay, now this slide tells us a little bit about specificity. Now, as was obvious, the T cell activation occurs via the T cell receptor, and the cognate ligand for the T cell receptor is the MHC peptide. Now, upon activation of that, you have T cell growth factors that are produced that results in proliferation of these T cells. Now, IL-2 and IL-4 is produced by T cells, whereas IL-15 is produced by macrophages. IL-15 is also an important factor which allows for T cells to proliferate. Now, how do we study T cell activation? If you just think a little bit about it, the T cell receptor is specific for MHC peptide. Now, the T cell receptor is variable, and so the chances of finding a particular antigen or cognate antigen is going to be very difficult. So, what researchers used were to use some non-specific mitogens initially. So, lectins, for example, concanavalin A is a lectin, it binds to T cell receptor and activates T cells. So, those were ones that were sort of used. Subsequently, what was found is that you could use pharmacological agents. So, for example, the combination of PMA and a calcium ionophore ionomycin, for example, would results in T cell activation. Now, singly these do not work, but the combination works, and this becomes very important because when we try and look at the T cell activation pathway, we find that there is one part where the formol ester activates the diacylglycerol pathway, resulting in and the PKC activation mode, and the other is calcium. So, these two combine to result in maximal activation of T cells, and this is something that we will study. PMA, for example, binds constitutively to protein kinase C and keeps it activated. And what this ionophore is doing is it opens up calcium channels, so you have increased amounts of intracellular calcium. Now, we had talked about antigens, which is antigens from MHC peptide, as being the most physiological. Now, however, there are some super antigens. Now, this antigen is very specific for the cognate T cell receptor. Now, you have super antigens. Now, super antigens will activate a large number of T cells having diverse TCR specificities. They may bind to families of T cell receptors, but overall they activate a large pool of T cells, and so that is why they are known as super antigens, because they are not the normal MHC peptide complex antigens. The other ways of activating T cells is to use antibodies to T cell receptor or CD3, which is you activate the antibody binds to a constant region of the T cell receptor, or it binds to the CD3 complex, and it induces T cell proliferation. Now, so that is how you can activate, but in order to study activation, you can take a look at surface expression. So, once T cells are activated, they express certain cell surface molecules, for example, CD25, CD69. These are expressed upon activation. So, you can look at the kinetics of expression of these molecules and find out what percentage of your cells are activated. In today's world, if, for example, you have a molecule that is important for T cell activation, so we need to use usually a knockout technology by which you have mice that don't express this particular molecule, and then see the effects on T cell activation. On the other hand, what people often use is TCR transgenic mice. So, you have mice that express a particular type of T cell receptor. So, you can now feed it antigen, and you can actually look at cognate TCR cognate antigen interactions, because you have majority of the T cells that would express this particular T cell receptor. So there are different ways of studying T cell activation. I hope this gives you a certain idea about the different ways T cell activation has been studied, and you also need to understand that it is somewhat difficult to study T cell activation, primarily for the reason that, especially because with respect to specificity, because the T cell TCR is specific. So there are ways by which we can bypass it. You can use molecules like lectins, which will bind to larger pools of which will bind to non-specifically to T cell receptors and activate them, or you can use super antigens which will bind to classes of T cells and activate them. The other way to activate, you bypass the surface interactions, is to use PMA ionomycin, and it results in non-specific activation of all types of T cells. T cell super antigens over here. So super antigens are ones that cross-link MHC class 2 and V beta. So these super antigens are specific for certain V betas. So they will bind. So it doesn't matter what the V alpha is, and doesn't matter what the antigen is. It will bind to MHC2 and the V beta family, a particular V beta family, as a result of which it will activate. This binding will result in activation of a pool of T cells, and there are two different types. One is the exogenous ones, that means they come from outside, and these would be examples would be bacterial toxins. So for example, the staphylococcal toxins, the staphylococcal toxic shock syndrome, TSST, and then you have staphylococcal enterotoxins. Then you have also endogenous ones. Endogenous ones are products from viruses that are within mice, or maybe are with humans, and they sort of get passed down. And this is known as the minor lymphocyte stimulatory antigens, and one of which is encoded by the mouse mammary tumor virus, MMTV, and so they were being actually passed down from a mother to litters, and it was being passed down. And since they are now seen as endogenous, so those mice lack certain V betas because they are seen as self, and so this deletion happens during thymic differentiation. Now this is what I was planning on saying, that the MLS antigens are present in the body from birth, and so therefore they are recognized as self and are deleted. Now an example of this is seen in the AKR mouse strain, where you have this endogenous mammary tumor virus, and it produces MLS1, and it is recognized by this by a large number of families of T cell receptor, the V beta 6, 7, 8.1 and 9, and therefore these TCRs are absent in the periphery, because they are seen as self and are deleted in the thymus during thymic differentiation. This is an example of antigen. This is, you have the MHC class 2, this is the T cell receptor, and here you see that the super antigen is being presented by the APC, and it is cross linking the MHC class 2 as well as the TCR beta part, and so it results in activation of the T cells. Now in terms of general T cell activation, what the way I look at it is, there are some general themes, and I think as long as students understand the themes, then they will understand the nitty-gritty details. So in terms of themes, you have first receptor ligand interactions, which is important in terms of specificity. Then you have phosphorylation, dephosphorylation events, followed by which you have recruitment of adapter molecules. And adapters are important because they can recruit other molecules that would amplify the signal. You have generation of second messengers, then you have the signal amplification, and then ultimately gene expression. In terms of T cell activation, the gene expression part is really comes down to the level of cytokines, and especially IL-2, which as mentioned is the autocrine growth factor. So we start off with TCR MHC and then end with IL-2. Okay. So and that is the important aspect, and this cartoon sort of depicts that. So here you have the TCR CD3 complex with the MHC peptide, it gets activated, and you can bypass this using anti-CD3. and then what we have is phosphorylation, dephosphorylation events leading to activation of the phosphorylation of the ZAP70 and LAT or linker associated in T cells. So, these two are adapter molecules, and subsequently you have what is an amplification of the signal where you have the PLC gamma getting activated. It results in activation of the diacylglycerol and the IP3. This results in increase in calcium, and you can see this activates a phosphatase known as calcineurin. This results in activation of a transcription factor known as NF80. Whereas the diacylglycerol part activates the protein kinase C, and this results in activation of the NF kappa B part, which again contributes to IL-2. You also have the activation of the RAS pathway or MEK pathway, and then you have the activator protein AP1 being produced. Now, what is important to note over here is that for IL-2, for optimal IL-2 production, you need the three important transcription factors to be induced and activated. The first one is NF80 or nuclear factor presented in activated T cells. You have NF kappa B, and then you have AP1. So, you have a signal right from here. It needs to have pathways that would activate these three. Now, if all the three pathways are not activated, what happens is you do not have proper T cell activation. And that is a very important aspect to understand. So, for example, if you do not have NF80, then again you do not generate IL-2, and the T cells will not get proper T cell activation. T cell activation will be compromised. The other important point to note over here, I understand it is a busy slide and we will break it down in the subsequent slides, but for now there are some important aspects for students to understand. One is this is an overview of T cell activation. So, it gives you a pretty much a bird's eye view of the different pathways that are involved in T cell activation. We will discuss these in greater detail. Second is there are ways by which you can bypass T cell activation. Here, for example, is the TCR MHC peptide is the most physiological cognate interaction. You can bypass it using anti-CD3, and if you do not have anti-CD3, you want to activate all types of T cells, you can bypass it by using the combination of forbolester PMA, which activates PKC constitutively, which binds and activates PKC constitutively. And the other way you need to do it is to open up the calcium or increase intracellular calcium, and this is a calcium ionophore. Ionomycin is a calcium ionophore, and which will result in activation of which will increase intracellular calcium. Again, what is important is that you need the combination of a calcium ionophore and PMA or forbolester to activate T cells. Again, singly they will not be able to do it. The third important point over here that is shown is that cyclosporine, with the immunosuppressant cyclosporine, the target is calcineurin, the phosphatase. Okay, these are important aspects. We will discuss this in greater detail in the subsequent slides, but you have a bird's eye view over here of pretty much of T cell activation. Once again, to remind you, you have T cell, you have activation of the T cell receptor. It needs to activate three independent pathways over here result in increased production of these three transcription factors and to induce IL-2. Okay, so now we will break it down a little bit. So, this is the T cell receptor, which the cognate the ligand is the MHC peptide, and the T cell receptor is associated with CD3, and you have the gamma, delta, epsilon, and here you have zeta. Now, what is important to note is that the gamma, delta, and epsilon have a single ITAM motif or immunotyrosine activation motif, whereas zeta has three activation motifs. So, this is important, and we will see that subsequently. What is also shown over here is that CD4, CD8 are present on T cells and associated with a molecule known as LCK. So, this is again something that is going to be important. We will discuss this later. Okay, so what I have shown over here is you have the TCR MHC. TCR is important for recognition, but the signal is actually passed down by the CD3, which is important for this aspect. Okay, now the activation now of CD3 gets phosphorylated. Now, for CD3 phosphorylation, you have the tyrosine kinases FIN and LCK to be important. Now, under normal circumstances they are inactive. they are inactive because they get phosphorylated by a kinase known as CSK or C-terminal Src kinase. So, as a result of which they are inactive and they cannot phosphorylate the ITAM motifs in the CD3. Now, however, upon T cell activation, what happens is you have CD4, CD45 being activated, CD45 then dephosphorylates LCK fin, which in turn results in increased phosphorylation of CD3 or increased phosphorylation of increased phosphorylation of CD3. So, now what happens is you have the ITAMs being phosphorylated. This allows a docking site for another protein known as ZAP70. ZAP70 stands for zeta associated protein, and this was found because only upon T cell activation it was found that the protein associates with zeta, and that was identified to be the ZAP70, and 70 stands for the KDA. So, it is a 70 KDA protein. Now, again as I mentioned, what happens with T cell activation is, once you have T cell activation, you need to bring it down. So, the cell as well as the host have figured out ways by which you can do that, and one important molecule over here is Cybil. Cybil is a E3 ubiquitin ligase which is important in and a substrate for Cybil is ZAP70. So, so what Cybil does, it will reduce activation of ZAP70 or reduce amounts of ZAP70. So, as a result of which you can lower T cell activation down. In conditions where there is no Cybil, what would happen? You would have increased phosphorylation of ZAP70, and that would go on and lead to problems with autoimmunity, and that is something that we will see later. So, once you have ZAP70 being phosphorylated, it recruits other adapter molecules, one of which is important one, which is LAT, which is linker in activated T cells, and SLP70. So, these all accumulate in a particular part in the T cell surface, and this part is known as the supramolecular activation cluster. So, what happens is over here you have the T cell receptor, the CD3 and other activation molecules all present over here, and there are high amounts of cholesterol over here, and so membrane fluidity is reduced. So, they are all present over there and they are bound to the MHC peptide complex, and so it results in increased signaling, and you have in fact molecules coming over here and they form this particular cluster known as the SMARCs. Once you have this, you would activate different pathways, and what is shown over here is you have activation of the RAS pathway. This results in activation of the MAP kinase pathway, increased CFOS, CJUN, and ultimate formation of AP1, which is one of the important transcription factors. So, AP1 is actually CJUN, phosphorylated, and CFOS. And over here you have the phosphoryl inositol bisphosphate being cleaved due to activation of PLC gamma 1. It results in diacyl glycerol. Diacyl glycerol binds to PKC theta, and in fact PKC theta is thought to be important in T cell activation. This activates it. It results in phosphorylation of the inhibitor of kappa B. Now, you will remember that the NF kappa B is bound to the inhibitor of kappa B in the cytosol. So, once the I kappa B gets phosphorylated, it gets degraded, and so now the NF kappa B is free to move to the nucleus and activate genes. And the IP3 increases in inositol trisphosphate increases calcium concentrations. It results in activation of calmodulin, and this results in activation of the calcineurin, which is a serine threonine phosphatase. Now, the cytosolic NF-AT is phosphorylated. What calcineurin does is to dephosphorylate it. Upon dephosphorylation, NFAT moves to the nucleus, and so now you have these three transcription factors AP1, NF kappa B, and NF-AT in the nucleus, and this results in increased gene expression, especially IL-2. So, again, to reinforce, for IL-2 to be activated optimally, it needs the three transcription factors AP1, NF kappa B, and NF-AT. So, this is again to show the different pathways that are involved. This is the ZAP70, and it allows for binding of the other molecules and the calcium pathway, and then you have all these different pathways coming together. It results in changes in gene expression, functional changes, differentiation, activation, so on. Now, gene expression in activated T cells occurs very quickly. one of the first ones to be activated within 15 minutes is the C4C gene. Subsequently, the cytokines get activated, IL-2, IF and gamma, by about 30 to 45 minutes, and then you have CD25 or the P55 IL-2 receptor, which gets increased by about 2 hours. So, IL-2, the CD25 is an important marker, a cell surface marker of T cell activation. IL-2 mRNA may be getting induced, but it takes some time for the protein to be made and be expressed on the cell surface. CD25 is also expressed very early, reasonably early in T cell activation, and is a marker of T cell activation. There are some late ones like HLA-DR, which is MHC class 2. In human cells, HLA-DR gets activated, and you see it present, it's induced by 3 to 5 days, and then you have other antigens known as the VLA4s or the very late antigens. We will now discuss some important molecules in the T cell activation. The first one is protein kinase C theta. Now, there are different protein kinases. The one that is thought to be physiologically important for T cell activation is the PKC theta, and this one is a calcium independent protein kinase family member. And the PKC theta knockout mice are deficient in T cell proliferation, cytokine production, although thymic differentiation does not appear to be affected. Now, a key feature of PKC theta is its ability to translocate to the SMARC, or which is where I said all these molecules form together along with high cholesterol during T cell activation, and this is sort of dependent upon some G protein like AVAV and the exchange factor RAC and CDC42. Now, calcium is really the important one very important component in T cell activation. There is, under normal circumstances, the amounts of intracellular calcium are about 100 nanomolar or so. Once T cells are activated, they rise very quickly to about 1 micromolar, and this initial rise in calcium is due to depletion from the endoplasmic reticulum, is released from the stores in the endoplasmic reticulum. However, for sustained T cell activation, you would need to have a rise in intracellular calcium. So, you need an increase in intracellular calcium, and which you need to be able to sustain it. If you are unable to sustain it, you will not get proper T cell activation. And so, therefore, following depletion of these stores, extracellular calcium of the depletion of the intracellular calcium stores, you have calcium coming in from outside to maintain high levels of calcium. High amounts of intracellular calcium are important because they are required to sustain the activation of NFAT, which is important for IL-2. And the calcium release activated calcium channels are important in this process, or the crack channels. There are two important players in this. You have ORI1 and STIM1, and ORI1 is a calcium release activated calcium channel. It contains 4 members and is present in the plasma membrane. So, it is a calcium channel, it is a proper calcium channel, it is present on the plasma membrane. And whereas STIM is mainly a sensor, it senses intracellular calcium, and when there are low amounts of intracellular calcium, there are conformational changes in STIM, leads to multimolarization and translocation to the membrane, to the plasma membrane, to activate ORI1. So, it is a sensor and an activator of ORI1. So, what would happen to people who either have deficient ORI1 or STIM1? And so, absence of these molecules leads to failure to activate NFAT and production of different types of cytokines in T cells. And these affected children are highly susceptible to recurrent infections at early age, and this results in aberrant T cell activation and will naturally have immune defects. Okay, so this is again to a cartoon to tell you a little bit about T cell activation. So, this is the TCR and it results in inositol trisphosphate, and this results in depletion from the intracellular in the ER calcium, it goes outside, and then these calciums, intracellular calcium activates the crack channels, and once crack channels are activated, you have now calcium coming in from the outside coming inside. Now you have sustained amounts of of calcium, which results in activation of calmodulin. Calmodulin on the other hand activates calcineurin, which is the phosphatase, and now the phosphatase, it cleaves the cytosolic NF80, and then it upon dephosphorylation NF80 migrates into the nucleus and it activates IL-2. So, this is again a cartoon to depict calcium channel signaling in T cells. An important aspect over there was the activation of calcineurin. What is interesting is cyclosporine, which is a very effective and useful immunosuppressant. I mean, cyclosporine has been used for quite some time as an immunosuppressant. However, the mechanism was not known, and now it is well known. What cyclosporine does, it binds to cyclophilin, and you have another molecule known as FK506. Now, FK506 binds to its cognate ligand, which is FK506 binding protein. It does not matter, both cyclosporine and FK506 are immunosuppressants. They bind to different proteins, but this drug and binding protein complex, it inhibits calcineurin. Now, it inhibits calcineurin, therefore, it would inhibit the production of IL-2, because what happens as a result of inhibition of calcineurin, you have the cytosolic NF80 cannot be dephosphorylated, as a result of which calcineurin remains in the cytosol. NF80 remains in the cytosol, and as a result it cannot translocate to the nucleus to activate IL-2 gene transcription. So, as a result of this, you have the inhibition of T cell activation. An important aspect over here is that the T cell receptor signaling pathway for IL-2 is sensitive to cyclosporine, whereas the CD28 pathway is resistant to cyclosporine. So, there are other ways by which CD28 acts, and this becomes important to differentiate whether a particular T cell activation is going primarily through the T cell receptor pathway, or it is going through the CD28 pathway or the co-stimulatory pathway. So, this is what is shown. You have calcium, calmodulin, it enhances calcineurin activity. So, you have more phosphatase activity, resulting in more translocation of the cytosolic N fat into the nucleus. So, you have more nuclear N fat. So, therefore, you have more IL-2. Now, what happens when you have cyclosporine cyclophilin complex, this inhibits calcineurin activity, as a result of which you have less nuclear NF80, you have less IL-2. So, this is important in terms of mechanism of cyclosporine action, which is very important. And even if you see some of the newer drugs that come out, these are all these pretty much act on this particular pathway, which is inhibition of calcineurin. So, they may have different binding proteins, or they may have slightly different structures, but the mechanism of action is inhibition of IL-2, inhibition of calcineurin, which leads to inhibition of IL-2, very important. Okay, so another aspect of T cell activation is T cell co-stimulation, and this the basis for this is as follows. You have I mentioned that you have the T cell and you have T cell receptor, and the T cell receptor recognize the MHC peptide complex on the APCs. Now, just this binding alone, while it is specific, this binding alone does not result in optimal T cell activation. In fact, it results in reduced T cell activation, T cell energy, and possibly death. Now, for optimal T cell activation, what you need is signal 2. And so, this is what is shown over here, signal 2 in the form of CD28. It is a cell surface molecule present on T cells. It binds to ligands on antigen presenting cells known as CD86. The binding of these two together results in optimal T cell activation. Why is it that you need to have these two signals to activate T cells? Why cannot just a single signal activate T cells? And so, this is perhaps because every time the T cell receptor sees a MHC like cognate molecule, if the interaction is not all that optimal, it should not activate T cells. So, you need T cell activation needs to be in a context where you have the conditions are right for optimal T cell activation, and we will discuss this part a little bit in greater detail. So, what happens is once you have this environment or inflammatory situations, you have the co-stimulatory ligands being expressed. Once you have these co-stimulatory ligands being expressed, they will bind to the co-stimulatory receptors, and if the MHC TCR interaction is there, only then will it activate T cells. And if you just have the signal 1 interaction, it is marked by inability of the cells to proliferate in response to the peptide MHC complex. So, this is this is shown here in this model, where if you have the calcium signal alone, it results in activation of NF-AT and it results in energy. So, this is why I said for optimal T cell activation you need different pathways coming together which would optimally activate your T cells. So, and if you have just one signal alone, for example, what is shown over here is a calcium signal alone, it results in activation and it results in energy, because you have NF-AT, but you do not have the other molecules. So, you need a combined input of different signals to tell the cell that look, now you know it needs to be activated properly. So, in fact, with NF-AT mice that lack NF-AT are less prone to T cell energy, and therefore, what is shown over here is activation of both NF-AT AP1 and NF-kappa B are required for optimal T cell activation. Okay. So, this is the question, why is this physiologically relevant? So, the T cell immune response is initiated during inflammation, and this helps prevent autoimmunity, because every time, if for every little thing the T cell saw something and then it got activated, it might result in autoimmunity. And this is not to say that autoimmunity exists in the populations, but perhaps those numbers would be lot more if this different signals or the co-stimulatory aspect was not required by T cells. You would have much more numbers perhaps of autoimmunity. And so T cell activation is done in terms of a context, and when upon the proper environmental cues. For example, the use of adjuvants, bacterial products, they increase inflammation. They will increase the surface expression of co-stimulatory ligands, and all of which would help in the immune response. Now, as mentioned, in some cases T cell activation, you have signal 1 and it results in energy. In these cases, the T cells cannot secrete IL-2 and proliferate. So, the biochemical basis for energy is still being understood. So, as was shown in that slide, an important co-stimulatory receptor that is present on T cells is CD28. Now, what is important is that CD28 is present on both resting as well as activated T cells, and CD28 binds to molecules known as B7, 1 and 2, also known as Cd8 T 86, and Cd8 T 86 are primarily found on antigen presenting cells. So, you have the T cell receptor CD28 which binds to the ligands in APCs. Now, remember the amounts of Cd8 T 86 increase upon inflammatory conditions. So and that is when the chances of co-stimulation are highest, and that would allow for T cell activation to occur under proper physiological conditions. Now, what is also shown over here is you have CD28, you have another molecule shown as CTLA4. Now, CTLA4 is not present on the surface of naive T cells. CTLA4 comes up upon T cell activation. And again, this is a mechanism by which CTLA4 competes with CD28 and it actually shuts down T cell activation. So, you have CD28 which enhances T cell activation along with the TCR, and then you have CTLA4 which binds to CD8 T 86 and but has the opposite role. So, why would you want that? Again, as mentioned to you, for T cell activation, once you have T cell activation, you would want molecules by which you can regulate this process and bring it down. It is not in the interest of the host to have sustained T cell activation, because then it would result in immunopathological conditions. So, this is a very important aspect for students to understand. A little bit about CD28. CD28 is a 44 kDa homodimer. And it is constitutively present on Cd4 positive T cells. Its ligands, as mentioned, is Cd8 T 86. CD28 is a positive co-stimulator of T cell activation. What is important is that the CD28 knockout mice can initiate, but cannot sustain T cell responses. So, they will initiate, but sustenance of T cell responses needs co-stimulation. And so, what are the mechanisms by which it enhances signaling by the various pathways, P38 MAP kinase, PI3K, so on. It increases production of IL-2 and other cytokines. This, and what is important is that it not only increases transcription, but it also increases the half lives of these cytokine mRNAs. This is a very important aspect. For CD28. The other very important role of CD28 is that it reduces the T cell death, or in other words, it increases T cell survival, and it does so by induction of anti-apoptotic molecules. BCL2, BCLXL are induced by CD28, very important aspect. So, it uses a combination of mechanisms. It activates T cells, but it increases the production of cytokine mRNAs, as well as increases the half life. So, you have more amounts of cytokines, and it increases T cell survival. So, the combination of these increases proliferation, increases survival. So, it plays a very important role in sustenance of T cell responses. Now, upon activation, what happens is CTLA4 now comes up. Now, CTLA4 is similar 43 kDa, it's expressed among T cells post activation. Now, it binds to CD80-86 just like CD28, but it does so with a higher affinity, and it prefers to bind CD8. What the phenotype of the knockout mice is striking. In fact, CTLA4 knockout mice die within 3 to 4 weeks, and they die because due to hyper proliferation of lymphocytes, and these are primarily CD4 positive lymphocytes. So, in the absence, so this is what is happening in the absence of CTLA4, you have the CD4 positive cells, but they are now hyperactivated. Any small little thing they are getting activated, and as a result of which they are producing a whole bunch of cytokines, they are activated, and it results in immunopathology, and so the host cannot handle this sort of a situation, and then ultimately the mice die. And so, because of the phenotype of these mice, CTLA4 is thought to be a negative regulator of T cell responses, very important aspect over here. Now, some of the mechanisms of CTLA4, so how does CTLA4 work? We had heard the mechanism by which CD28 works. It increases anti-apoptotic molecules, it increases production of cytokines, but CTLA4 has a different mechanisms. Due to higher binding affinity, it can outcompete CD28 for binding these ligands. It also there are different mechanisms listed, and I will tell these mechanisms, and then we will try. Perhaps a combination of these mechanisms are effective. It associates with the phosphatase, so as a result of which it down modulates T cell activation. It decreases MAP kinase NF80 activation, resulting in decreased IL-2 T cells cycling. It increases IDO expression. What IDO will do, it results, and as a result of which you have less tryptophan, which is essential for T cell proliferation. Also, it prevents prolonged interaction between T cells and APC. So that prolonged interaction of the SMARCs that we discussed, that is reduced with CTLA4. So, perhaps there is less interaction, so less T cell activation. And so, inhibition of T cell responses, we had discussed this aspect, that there is a need to down modulate T cell responses, because otherwise the patient may die due to uncontrolled immune responses, and this is what I mentioned by immunopathology. And a good example of this is CTLA4, which downregulates T cell activation. So, actually in this class we started talking about T cell activation. We looked at the different pathways, different molecules that are important in T cell activation, and finally we are actually coming down to inhibition of T cell responses. And because whatever, if you activate immune cells, you need to find out ways by which you can bring them down, because having activated immune cells is not in the interest of the host. One of the mechanisms that we discussed in this class regarding inhibition of T cell responses is CTLA4. There are other mechanisms, but I thought for an introductory class on T cell activation, we will discuss the simple one, and CTLA4 is a very good example of the importance of regulating T cell activation. In fact, this interrelationship between CD28 and its cousin CTLA4, because remember they are cousins because they are related to each other. They bind to the same ligands, but they have completely opposite functions. CD28 is a positive regulator, CTLA4 is a negative regulator, and the mechanisms by which they function are also very distinct, and they have differential effects on T cell activation. We will now briefly summarize this class. So, first is what is the importance of studying T cell activation. We discussed this. T cell activation is important in terms, especially in terms of transplants. You want to be able to suppress, so that you give some time for the graft to be accepted by the host. On the other hand, in case of vaccines, you want to induce T cell activation. There are different methods of studying T cell activation. Now, in most methods, what people use is the use of antibodies to send a signal through the T cell receptor. You can use anti-TCR or anti-CD3. Anti-CD3 is more commonly used. There are different pathways that are involved. We discussed the whole gamut of pathways, right from the role of SYK in keeping the LCK in an inactive form, and once you have CD45 being activated, it dephosphorylates LCK, and then which now activates the items get phosphorylated, and once the items are phosphorylated, you have ZAP70 being recruited over there. ZAP70 in turn is a tyrosine kinase, it recruits other ones. We also importantly discussed the role of Cybil. Now, Cybil is a E3 ubiquitin ligase, and one of the substrates for Cybil is ZAP70. So, in fact, in mice that lack Cybil, you have increased ZAP70 because there are the enzymes to bring down ZAP70 are not that efficient in the and as a result of which you have again hyperactivation of T cells, and which gives you an autoimmune phenotype. So, this tells you about these very important regulators of T cell activation. Then you have we also discussed the mitogen activated the MAPK phosphorylation pathway, and the ERK, for example, is a very important. The phosphorylation of ERK is an important aspect over here. These finally lead to the activation of the three important transcription factors, NF-AT, NF-kappa B, AP1, leading to IL-2, and IL-2 is the autocrine growth factor. So, that is where it sort of comes down to. So, you start off with the T cell receptor and you end with IL-2, and in this intermediate you have all these different players, especially the three transcription factors and leading to IL-2 synthesis. What is emphasized, or what was emphasized over here, is that if you just have a single transcription factor being affected, it does not lead to proper T cell activation. It can have negative consequences. For example, calcium alone increases NF-AT, but what it does is that it makes the cells anergic, which means they are now resistant to T cell activation and T cell activation and proliferation. We also discussed the role of calcium and T cell activation in this process. Calcium is extremely important. You have an initial burst in calcium and then followed by sustenance of intracellular calcium. In the absence of sustenance, you do not get proper activation of calcineurin, as a result of which the cytoplasmic NF-AT is not dephosphorylated, it remains there, and you do it, and it cannot translocate to the nucleus, as a result of which you do not have enough IL-2 being activated. The other important aspect that we discussed was the mechanism of cyclosporine action and the immunosuppressants. Very important. Cyclosporine binds to cyclophilin, and this complex binds to and inhibits calcineurin, which is the phosphatase. I think the mechanism of action of cyclosporine is very important, and students should pay very careful attention to it. These are important because of the translational benefits that it has. Finally, we discussed the role of T cell co-stimulation, especially the role of the cousins CD28 and CTLA4, one being a positive co-stimulator, one is a negative stimulator. They bind to the same molecules, but they have different functions, their expression is different, their mechanism of action is different. So, overall this class was on T cell activation. So, I hope you understand the importance of T cell activation and the pathways and the translational benefits of T cell activation. Thank you.